Hi guys. Well, I am recording from home today, so let's see how this goes. It's usually pretty chaotic here, and I'm sure it's going to be as we go through this. So, bear with me and my crazy little family. All right. So, we're on chapter 6. And we're talking about metabolism, energy, and enzymes. And the biggest idea that we're going to be talking about today is the enzymes idea. Okay, so we're going to spend a lot of time talking about enzymes. So before we get started, you should know the definition of this really important word, which is metabolism. So everybody, please say metabolism. Metabolism is the total of all chemical reactions that are going on in your body and in your cells. So you can have cellular metabolism, and that would be the metabolism of a single cell, or you can have your body's metabolism, which is the metabolism of all of the all of the cells in your body. I'm having such struggle, guys. This is the 4th time I've had to plug in my laptop. I'm sitting on the ground. Cannot record from home very often. It just doesn't work. Okay, so let's move on to the next slide here. So this is a typical metabolic pathway. A metabolic pathway shows the movement from the starting molecule to the product. Usually when it comes to chemical reactions in your cells, it's not just a simple one-step reaction. Usually in your cells, to go from the starting molecule to the product that you want, you've got multiple reactions to get there. And so this is called the metabolic pathway. And so you'll look, we start with this molecule and we need to get this molecule, but there's 3 enzymes or 3 chemical reactions that are involved with creating the product from the starting molecule. And so usually enzyme 1 will do one reaction, enzyme 2 will do a second reaction, and so on. And you can have so many different steps along this pathway, or a few steps on this pathway. It just depends on the reactions that we got to do. There's 2 main types of metabolic reactions that we have to know about. Star them in your notes. We will be talking about them a lot. And they actually get multiple names in here. So yay for multiple names. So first type of chemical reaction that we're going to talk about is an anabolic reaction. Everybody say anabolic. Anabolic are building reactions. So we build molecules. So if we were creating a bigger molecule from smaller molecules. And anabolic reactions cost energy. It takes energy to build things. The opposite of that, seriously, the opposite of that is catabolic. So everybody please say catabolic. Catabolic. Okay, catabolic is where we break things down. And when we do catabolic reactions, we release energy. So those are our 2 main reactions. And by releasing energy, we basically mean when we break the bond holding 2 molecules together, we release the energy that's held in that bond. All right? And energy and metabolism are intertwined. Teddy, you want to say hi? Come say hi to my students, Teddy.
Hi.
That's Teddy. He's 3, and he's my buddy. And this is Roland. He's 6, and he's my other buddy. And now they're going to leave me alone. Huh, guys? Okay, so energy is very intertwined with chemical reactions. Chemical reactions and energy, out, go on, require energy and release energy. And so this is just showing some of the released energy that's coming from a chemical reaction. These fungi are doing chemical reactions, and they're doing these wasteful chemical reactions on purpose so that they can release the light energy that's held within the bonds of the chemicals that they're reacting together. And so they're phosphorylating by releasing a small amount of energy by breaking down certain chemicals. Please, guys, go on. Teddy, you need to go in the other room. Okay, so here's a question for you. Which is the most abundant form of energy that I can find in a cell? Chemical and electrical gradients, heat energy, mechanical energy, chemical energy, or kinetic energy? I bet you could eliminate a few answers just off the top, and then go ahead and decide what you think the actual answer is. And ready for this? Maybe. Oh. It's chemical energy, like ATP energy, energy held within bonds, energy held within chemical bonds. All right, so let's talk about free energy. Free energy is the energy that is available to do work. And cells have to have a certain amount of free energy to do chemical reactions. They have to have a certain amount of energy available to do work. And this is our free energy equation. Guys, you will probably never use this equation in this class. I'm just going to throw that out there. I mean, there might be some reasons, but you're probably never going to input and actually use this energy because of some of the things are really hard to calculate and kind of way past the scope of this class. So free energy is equal to enthalpy minus temperature times entropy. Like, that's really out there kinds of stuff you talk about in chemistry and physics quite a bit. So if you have a chemistry and physics background, you might have used this formula, but not something we talk a ton about in biology unless you really are getting into energetics. But you will need to know this change in G a lot. So we're going to talk about that. But before we get started, let's talk about entropy. Entropy is something that I remember talking about in high school, especially in physics. So do you know what entropy is? Do you remember learning about entropy in physics or other classes? Where have we seen it before? So let's talk about entropy. All right. Everybody, please say entropy. Entropy. Entropy is the general idea of how spontaneous reactions will favor the disorder. That means reactions that happen by themselves favor the breaking of things down, favor disorder. Things go from organized to disorganized by themselves. It doesn't take any energy to go from organized to disorganized, but it does take energy to reorganize things, right? So if I were to have to, I mean, it didn't take much energy for this building to kind of fall apart. It just happens, right? It happens by itself. Things just happen. Same with my bedroom, right? That's not my bedroom, promise. Things just happen and it goes disorderly, and I have to put energy in if I need to reorder these things or rebuild these things. And that's what entropy is. In the universe, reactions tend to favor the disorder of the universe, and we have to have a constant supply of energy to maintain order. And this actually matters a lot to you and biology because cells are ordered. They are ordered. And so you have to put a constant amount of energy to keep your cells going and keep your cells built and keep your cells alive and keep your cells organized. So that's why you always have to have a constant energy source. So this is examples of entropy in living systems. So you might want to have a section in your notes about examples of entropy. The first one is that catabolic reactions will increase entropy. So catabolic reactions, remember, are breaking reactions. So here's glucose. And when we break glucose during respiration, we get water and carbon dioxide. And you'll notice we went from ordered to disordered, organized to unorganized, to random. When you break things down, you break them and you go from organized. This is an organized chemical, glucose, to disorganized, and these guys are free to move however they want. It's very random. So it's an increase in randomness because we're no longer have this ordered structure. Diffusion also increases entropy because we go from high concentrations to equal concentrations to random concentrations, right? So when these things move, they go from ordered to unordered. And when they do that, they release the chemical energy that does that. So once again, we have an increase in randomness because each ion has a choice of two possible locations that it could be. Here, it was in one spot. Here, it could be in any spot, back and forth, back and forth, back and forth, right? So diffusion will increase entropy too, from organized to more disorganized. A very important form of random energy in our cells is heat. Okay, so we need to know that a random kind of energy is heat. Heat energy is high entropy. It's very disorganized. So this is usable energy. This is very usable, organized energy. This would be like energy from the sun, right? Rays of light. This is unusable energy. This is heat energy. Heat energy is very random. Now, humans, we've figured out ways to use heat energy, like to cook things and stuff like that. And sometimes our cells can use heat energy, but not often. It's very hard to use this heat energy. This is unusable energy because it's so disorganized and so random. If our cells are using a source of energy, usually it's a very ordered kind of energy. Okay, most cells could not harness heat energy to perform work because A, heat is not a form of energy. B, cells do not have much heat. They are relatively cool. C, temperature is usually uniform throughout a cell. D, heat could never be used to do work. That's a lie, I'm just throwing that out there. And E, heat denatures enzymes. So why can cells not usually use heat to do work? It's because temperature is usually uniform throughout a cell. There's not a difference in temperatures between one part of a cell and another part of the cell. They're just too close together. Heat tends to move too much. If the entropy of an organism is decreasing, so entropy is decreasing, think of it, what must be happening simultaneously? If entropy of an organism is decreasing, the entropy of the organism's environment must also be decreasing. Heat is being used by the organism as a source of energy. The first law of thermodynamics is being violated, or energy input into the organism must be occurring in order to drive the decrease in entropy. So if you think about it, a decrease in entropy, if entropy is an increase in randomness, a decrease in entropy is an increase in order. It's kind of weird, right? And if we're going to increase order, that means we must be putting energy into that system. Okay, now, important. No energy conversion is 100% efficient. Every time we go from one form of energy to another form of energy, we lose some of that as heat. We lose some of that as random energy, and it just leaves the system. Heat is mostly unusable energy for living things, so it just kind of leaves the system and kind of disperses. So photosynthesis converts solar energy to energy in food. We lose quite a bit of heat energy that way. It's not efficient at all. In fact, it's really inefficient. Plants are actually pretty bad at it, but there's so much solar energy that they're fine. They'll do fine. And then when organisms eat that food and they convert the energy from their food to ATP energy, we lose some of that energy as heat. It's lost because it's just not that efficient. Living things are not that efficient. And even in non-living things, energy conversions are not 100% efficient. So good to know that every time you do chemical reactions, you lose some of your energy involved as heat energy. Switch. Come on, go to the next slide. Oh, and by the way, this is the second law of thermodynamics. Very important. Write it down. Okay. Basically, every energy transformation will increase the entropy of the universe because heat is always created during energy exchanges. And so every time you do an energy conversion, you increase disorder by adding extra random heat energy to the universe because we're just not that efficient. Okay, so let's go back to our Gibbs free energy equation. Alrighty. So this is what I need you to know about this equation. It's not that awful. You don't have to solve or anything, but here's what I need you to know. Okay, if there's a positive change in G, that means it's an unfavorable reaction because energy was put in. Okay, we need to put energy in. That's an unfavorable reaction. So if this is positive, then it's unfavorable and it's going to have to put energy in. That means it's anabolic, by the way, because we have to put energy in. If we have a negative change in G, so if this number is negative, that means it's a favorable reaction, and that means energy will be released, which, by the way, means it'll be a catabolic reaction, right? So in biological systems, because of the second law of thermodynamics, living things have to follow the second law of thermodynamics. All reactions must be overall favorable. So overall, in your body, you have to have a negative change in G because that is the second law of thermodynamics. We must increase entropy. Okay, so we always have to have a negative change in G overall. Now, what happens if a cell does not have enough energy to maintain the building and the breaking reactions? Well, if we don't get enough energy, we're going to be unable to maintain order and we're going to die because things will just fall apart. And cells have to keep building and putting things together, right? And that's a positive change in G. So somehow we have to get enough energy to make up for that. Okay, so let's do this. So here's some important vocabulary we need to know. It will help us kind of get through this, okay? So, and this is related to your anabolic, catabolic, by the way. So you might want to put this around those kinds of notes. That might help. Okay, first off, there are exergonic reactions. Okay, exergonic, X means out, right? Out. So we're going to release energy to the outside. That's exergonic, okay? And these reactions are usually catabolic because we're breaking things and releasing the energy that you have in the chemical bonds. Okay, so these are catabolic reactions. And then there's endergonic. Ender means in, okay? So we're going to absorb energy inside, okay? We're going to take energy in. So endergonic reactions absorb energy. Now, these are usually anabolic because we need to absorb energy to build things, to build chemical bonds. So you probably want to have these graphs because they do help you see that change in G. So if you look, this is an exergonic reaction. Remember, that means we release energy. So here we have what we start with the chemical reaction. Here's how much energy we start with. Here's how much change in G we have, okay? So we start with this much G, this much free energy. Then we do the chemical reaction and we end up with a negative change in G. We lose free energy. See, we go from a more positive free energy to a more negative free energy. So we lose that energy and we release that energy to the outside world, usually as heat. So it's usually heat energy, but sometimes we can use this to make ATP and stuff, okay? So we have a negative change in G overall. We go from a higher change in G to a lower change in G. Or we go from a higher G to a lower G. Endergonic reactions are the opposite. Notice we start with less energy in our reactants than our products. So this means we have to put energy into this system in order to build the products. And that requires a positive change in G to do that. Okay? So this is an example of your catabolic reactions. Okay, these are our breaking reactions. These are our energy releasing reactions. We start with this much energy in our reactants. We do the chemical reactions. We break some bonds and we release the energy in those bonds. Some of that energy can be used to do work. Some of that energy gets lost as heat. And this is our product. So we have a negative change in G because we lose energy in those chemical bonds. Endergonic reactions, though, energy is required. So here, we start with our reactants here. And so now we're going to put some energy in to build something, put some energy in to build those bonds. And our products have more energy held within the bonds than what they started with. And so we had to put some energy in to build. So that's a positive change in G. But here's a question. How can we get anabolic, the building reactions, if they are endergonic and require free energy? In other words, where does this energy come from? How do we get this input? And that's kind of a really good question. And the way we're going to do this is we're going to do this with something called energy coupling. Okay? This is important. You might want to star it. Energy coupling is where we use the energy released from favorable reactions to drive unfavorable reactions. So we're going to couple these two reactions. We're going to break something down and release its energy, and then we're going to use that energy to drive another reaction. So you can break something and use the energy release to drive a different reaction to build. And that's called energy coupling. So it's kind of like, you can think of it like this in form of gears kind of thing. We break something and we get some energy. So we break that, and that energy is going to drive a building reaction. So we break something down to release the energy, and that drives a building reaction. So one of our more important favorable reactions includes the hydrolysis of ATP. That just means breaking down ATP, right? Because that's what hydrolysis is breaking things down. So remember, ATP is adenosine triphosphate. And one of the ways that we get a lot of energy, so one of these negative change in G reactions, is we can break ATP. And this bond holds so much energy that when you break this, it drives the other reactions. All right? And so that, we're going to talk a lot more about ATP later on and stuff, but that is kind of how that works. Maybe. Okay, so here we go. I told you we were going to talk more about ATP. I just didn't know it was so soon, which I don't know why. All right, so let's talk ATP. Everybody please say ATP. All right. Thanks, Teddy. Teddy said ATP. ATP stands for adenosine triphosphate, and it's really irritating to me that I have to write with this because my hand really hurts today with my cute little hand surgery. So I'm going to try and find adenosine triphosphate for you so that I don't have to write it down because my hand hurts too much. So adenosine, this is how you spell adenosine triphosphate, by the way, because I'm too lazy. So adenosine phosphate. There we go. Or ATP. ATP. Adenosine triphosphate. Go play, Teddy. Okay, so this is adenosine triphosphate, and the energy in ATP is held within these bonds. Okay, so it's held within these bonds, the phosphate bonds, and I like to think of this as stretching a spring. So I want you to think that I have, and this is my room. Here's my desk. Here's my desk. Oh my gosh, I'm really bad at that. It's opposite. Here's my desk. And then here's my lab bench. And I'm going to stretch a string from here all the way over to here. And I'm going to connect the spring between these 2 points. When I do that, when I stretch and I build and I connect these 2 points, that harvests potential energy, right? When I shove this guy to that guy, that puts some energy into that bond. Okay, so that's a building reaction. You have to put energy in to shove these two together. But when I break this bond, I'm going to release that potential energy, and I can use that energy to do work. So that is kind of how ATP works. And so this is the hydrolysis of ATP, the breaking of ATP. If you put a water in there, because remember what hydrolysis reactions are. You use water to break bonds, right? So I break ATP down, I break this bond, and when I break that bond, I'm going to release all of that energy in that bond. And then I end up with ADP, or adenosine diphosphate, okay, plus a phosphate group. Now, if I want to build more ATP, I can. I just got to shove that guy on there. But it takes energy to do that, okay? So we'll talk about how we get that energy a lot later on. So more of this energy coupling. So this is an endergonic reaction. It's got a positive change in G. It won't happen by itself because we're going to have to put energy in to build glutamine. We got to take glutamic acid and ammonia, shove them together. That's got a positive change in G. That's not going to happen by itself. Positive 3.4 kilocalories per mole. So in order to drive this reaction, we'll couple them together. Hold on. I want to see how much more I have on the slide before. Okay. So in order to drive this reaction, we'll couple them together. What we'll do is we're going to break ATP using water. And when we break ATP, that gives us a negative 7.3. I'm getting a phone call from Midvale, Utah. Hold on. I'm just going to pause my presentation really quick. Hello? Sorry, I can't hear you.
Hi, is this Brooke?
Yep, this is her.
This is Taylorsville Physical Therapy.
Hi.
How are you?
I'm good. How are you?
Hi. Good. I was wondering.
Sorry about that, guys. I had to answer that. So now I'll go back to this. Okay. So back to energy coupling. So remember, we're going to, in order to build this, we've got to get some energy. We got to get a positive change in, we've got to get some G, right? Some energy. So in order to do this, we're going to break this bond and we're going to release this much energy. Notice the negative change in G. That's an energy releasing reaction. So we're going to do the ATP reaction to turn the crank to drive building this. So we'll use the energy released here to do this reaction. And remember, we always have to have an overall negative change in G because we have to increase the entropy of the universe. And so if you see, this reaction provides plenty of energy to do this reaction, and we even have some left over, and this will usually be released as heat energy, okay? So here's another actual biology example of this reaction, which is kind of cool. It's phosphorylation. Everybody please say phosphorylation. This is really important, so make sure you write this down, okay? Phosphorylation is where we make something unstable, make something reactive by adding a phosphate group. Because if you add a phosphate group, it makes things unstable and makes them do things that wouldn't happen by themselves. So let's see how this works. So this is how that ATP hydrolysis, that energy coupling actually works. So what we want to do is we want to get this motor protein to take a step, right? We want to make it unstable enough to get it to move this vesicle forward a little bit. So to do this, what we can do is we add a phosphate. It makes it a little unstable. We're adding a phosphate, making it a little unstable so that it takes a step. So the way we add that phosphate, though, is we break ATP and we throw that phosphate onto him. So normally this guy wouldn't walk. It would just stay by himself until we add the phosphate. Adding that phosphate by breaking the ATP makes him unstable enough that he takes a step, okay? And then if we wanted to make him do another step, we'd have to add another phosphate to make them unstable enough to take a step. Same thing here. If we want this membrane protein to change shape, to move this stuff from inside to outside, we're going to add a phosphate by breaking the ATP. Breaking the ATP, we add that phosphate to that enzyme. It makes it unstable enough to take a step or to open up and do the other thing. And here's that reaction with glutamic acid to make glutamine. You break the ATP, you add that phosphate. It makes this guy unstable enough that they just shove themselves together. Adding that phosphate makes things unstable and makes things happen that wouldn't happen by themselves. Which brings us to the ATP cycle. Okay, so this is just kind of the same thing. This is a cycle constantly going on in your cells. Because you can see we have to have a constant supply of ATP in order to do work in our cells. So we do that by breaking down our food from exergonic reactions. This is respiration. This is breaking down our food. When we break down our food, we use the energy from that to shove these guys together to make ATP. And then when we need to do work, we break ATP to do cellular work, right? And use the energy held in ATP, and we end up with ADP plus phosphate. And so if we need to make more ATP, we shove them together. So that's our ATP cycle. And our main reactions for this, we get this energy from our food. So energy from our food, macromolecules that store the energy in our bond, carbohydrates, can be used to operate and build ATP. So when we break down our food, we release energy, right? So this is glucose. We release the energy found in glucose by breaking glucose. That energy is going to be used to make lots of ATP for our cells. And then we can use that ATP to do things, to build parts of our cells, right? And notice how it's really inefficient. We're really bad at getting energy from our food. We lose 60% of our energy as heat when we break down our food. We are so bad at it. We just eat so much that we're fine. And so this, remember, is lost as heat. Our reactions are not efficient at all. Okay, here's a question for you. Which of the following metabolic processes can occur without a net influx of energy from some other process? So which one of these can happen without energy being input? Can we go from ADP to ATP? Glucose breaking down to carbon dioxide and water? Carbon dioxide and oxygen being used to make glucose and oxygen? That's not even a reaction. And honestly, neither is that one. These reactions are stupid. Using amino acids to build protein. Using glucose and fructose to build sucrose. So which one of these reactions could happen without an input of energy? That would be B for sure, because B is a breaking reaction. I break glucose to make carbon dioxide and water. So this would not require an energy source because I'm breaking. In general, the hydrolysis of ATP drives chemical reactions by A, changing ADP, changing to ADP and phosphate. B, releasing energy, which can be used to drive other reactions. C, releasing heat. D, acting as a catalyst. Or E, lowering the energy demands of the reaction. So which one of these? Why does breaking down ATP drive chemical reactions? It's because it releases the energy that can be used. Okay. Identify this type of reaction. Is this an endergonic or is this an exergonic reaction? So looking at that graph, is it endergonic? Is it absorbing energy? Or is it exergonic? Is it releasing energy? That is a exergonic reaction because I start with a more positive change in G and I lose energy. So I have a negative change in G, and this would be an energy releasing reaction. All right. This is a good place to take a break because we've been going for a while. But this is where we kind of shift gears and talk about awesome enzymes. So if you need a pause and take a break here, I need you to be really fresh for this stuff, okay? So I'm going to go on. But like I said, if you need to take a break, go for it because enzymes are incredibly important. Okay, let's talk about enzymes. Enzymes are proteins that make chemical reactions happen faster. I wish I had my board here. Also, my hand really hurts today, so I'm doing my best, okay? They catalyze. Catalyze means to speed up, okay, to make things happen faster. So I always say enzymes are proteins that make chemical reactions happen faster. I want you to memorize that and say that to yourself before you go to bed. Enzymes are proteins that make chemical reactions happen faster. That's what they do. They're very important. So here is an enzyme that is doing an important chemical reaction. It's breaking sucrose down into glucose and fructose. So we're going to break it. And so this wouldn't happen by itself, really. It really wouldn't happen by itself without an enzyme. An enzyme is going to help break that and make that happen faster. So let's talk about the role of enzymes in a reaction. Now we're going to talk about specifically activation energy. So everybody please say, and star this is important. Please say activation energy or energy of activation. That's the EA, energy of activation, activation energy. This is my activation energy. Could be helpful to draw this in there if you need to. This is my activation energy. This is an energy barrier that has to be put into the system for the reaction to occur. This is how I usually put it, by the way. The energy required to get a reaction started. That's my activation energy. So I want you to notice the reaction is actually exergonic. This is actually an energy releasing reaction. I lose energy in this reaction. But you'll notice there's an energy input, an energy required to get the reaction started. That's my activation energy. I have to put this energy in to get this reaction to go, even though overall this reaction releases energy, right? Because I start here and I end up here. So even though this reaction can release energy, I still have to put a little bit of energy in to get them unstable to get the reaction started, okay? So I want you to draw this graph. This is a really important graph for you. You need to have this graph, okay? So what happens is, here is the reaction without an enzyme. Notice how much energy is required to get the reaction started. I've got to put that energy in. So this is the activation energy. But I want you to notice what happens if I add an enzyme. Notice the activation energy has been lowered. I need less of an energy input to get that reaction started. So this is the key thing about enzymes. Enzymes lower activation energy. They lower the amount of energy to get the reaction started. And that way, they can make them happen faster. Our cells would be unable to get this much energy ready to do a chemical reaction by themselves. They need enzymes to get this going faster. If I had to wait for my cell to get this much energy in, the reaction probably wouldn't ever happen by itself, or at least it would happen so slow that our cells would die while they're waiting for this reaction to get enough energy to go. So enzymes lower this activation energy enough to get it to go fast enough for life to happen, which is good. We need that to happen. And the way they do this is through something called an induced fit. Okay? So enzyme activity is all about induced fit. Here's what goes in. Here's what I need to break. Here's what I need to do. This is the chemical reaction that needs to happen. This substrate is going to fit into this active site just perfectly. And what happens is, when it fits in there, this enzyme makes it unstable enough that the reaction can happen faster. Okay, so it adds a phosphate group maybe, or it just puts things close together enough that it's unstable enough that it can do the chemical reaction. So the induced fit goes in just perfectly, okay? And this is the substrate is the reactant in the enzyme reaction, okay? So here we go. Here's how enzymes work. Substrates are going to enter into the active site. So these are my substrates. They enter into the active site of the enzyme through induced fit. So it's a fit that works together, right? And this substrate is held in by very weak interactions, maybe hydrophobic, hydrophilic interactions, hydrogen bonds, ionic bonds. So some very weak reactions are holding this in place. But it makes it unstable. This enzyme is going to lower the activation energy. It stresses bonds by keeping things so close together. It keeps things in the right orientation to allow them to be shaped well enough to do the chemical reaction. It provides a very stable microenvironment to allow that reaction to proceed faster at lower temperatures and at an unstable enough that the reaction can go, okay? And so when that happens, the reaction happened with less energy required, less activation energy, and the substrates get converted into the products. The products are released, and the active site is available for new substrates. And this enzyme can do it again and again and again and again. But this just creates a microenvironment unstable enough for these reactants that they're able to change into the products at a lower energy or with lower energy. Now, we're going to talk about some really important factors that influence enzyme activity. I would definitely make sure you have a list of these going on, okay? So anything that changes the shape of the active site is going to influence how well your enzyme does its job, Okay, so enzymes work by shape. Proteins work by shape. Biology works by shape. Structure determines function. Okay, it's really important. So if we change the shape of the active site, it might not be able to do its job, or sometimes it's able to do its job better. So this is an example of that. These are cofactors. Cofactors are helper pieces that bind to the active site and help it do its job better, okay? And that alter its shape. So here we have an inactive protein. It's not the right shape. It can't do its job yet. But if we add the cofactor, suddenly it has the correct shape that it can do its reaction and can convert these into something else, okay? So it needs a cofactor to do its job. Think of it like this guy turns the protein on. Without this guy, the protein doesn't work. The enzyme doesn't work. Cofactors are usually your metal atoms or ions, so iron, magnesium. Those are our cofactors. So your red blood cells, the hemoglobin won't work without iron. That's a cofactor that will help grab onto oxygen. There's also coenzymes, which are organic cofactors like vitamins, so vitamin B12 and stuff like that. Those might be cofactors that help turn your enzymes on, and why you need your vitamins and your minerals, right? Okay. Some other factors that influence enzyme activity are temperature and pH. Enzymes work at an appropriate temperature. Our enzymes work at about 37 degrees Celsius. That's where human enzymes work the best. Any guess on why human enzymes work the best at about 37 degrees Celsius? Why would my enzymes work the best at 37 degrees Celsius? 37 degrees Celsius is 98.6 degrees Fahrenheit. So it makes sense that our human enzymes work the best at human body temperature. Turns out if you get too cold, your enzymes start to slow down. So you start losing your body temperature, your enzymes start to slow down, which is why we like to stay at this internal temperature. That's why we're warm-blooded. If our enzymes get too warm, so our body gets too warm, it actually starts to break our enzymes down and they stop working pretty quickly. So they have an optimal temperature that they work at. But other organisms that have other optimal temperatures, their enzymes work better at those different temperatures. Like this is an organism that might live in a hot spring, like Yellowstone National Park. They live in a hot spring. They live in hot temperatures, almost boiling temperatures. So it makes sense that their enzymes would work best there. Our enzymes would die there. Our enzymes would not work at boiling. But once again, their enzymes won't work at our temperature. So we are evolved for the temperatures. Our enzymes are evolved for the temperatures that we live in. pH is the same thing. Our enzymes are evolved to work at the correct pHs. So in our stomach, our pH is like 2. And so our stomach enzymes work best at pH 2. Surprise! But our intestines aren't at pH 2. Our intestines are actually more at pH 8. So the intestinal enzymes, they work better at this pH. Makes sense, right? So enzymes are influenced by temperature and they're influenced by pH. Okay, so I want you to think where in your body you might find the following enzymes. So here's pepsin, here's carbonic anhydrase, and here is trypsin. So I want you to think, where do you think, based on what we talked about here, where do you think we find pepsin in our bodies? Pepsin is a stomach protein. It works in our stomach to help break down proteins, but it only works at a pH 2, right? Makes sense because it's a stomach protein. Trypsin, like you might have guessed, is a intestinal protein. It works in our intestines. Carbonic anhydrase works in your blood. Your blood pH sits right in between 6 and 7. Cool, right? All right, let's keep going. Other factors that influence enzyme activity. Okay, are enzyme inhibitors. Okay, so inhibitors are chemicals that can decrease enzyme function. They inhibit the enzymes. Oh, my butt is sore because I have been sitting on the floor. Anywhere else I sit doesn't work. I just gotta get up for a second. I'm gonna sit on my tummy. Sorry guys, I'm stretching for a second because my butt really hurts. Oh, sleepy butt. Okay, so inhibitors. Here is maybe a normal enzyme doing its normal job, right? The substrate fits right there. It's able to convert this guy to whatever it needs to convert it to. Now, this is a competitive inhibitor. Okay, so this is a type of inhibitor that you need to know. A competitive inhibitor competes for the active site. So this is the active site. This inhibitor just sat there and it blocked it. So this The substrate could not bind to the enzyme because of the competitive inhibitor that got in the way. It inhibited the protein. Now, there's also non-competitive inhibitors, and these are really important ones too. Non-competitive inhibitors still inhibit the enzyme, but they don't bind to the active site. They bind to a different site. When they bind to that, it changes the shape of the enzyme and changes the shape of the active site enough that the substrate can't fit. So it's a non-competitive inhibitor because it still inhibits the enzyme, but it's not going to compete for the active site like the competitive inhibitor did. So what do you think? What kind of bonds do you think are involved with these inhibitors? Do you think they're strong bonds or weak bonds, hydrogen bonds, ionic bonds, covalent bonds? And it probably depends on the inhibitor and the enzyme, and how long the enzyme is going to be infected or affected by the inhibitor. So this is just showing us the same thing, really important, okay? This is the normal enzyme substrate binding. Competitive inhibitors are blocking the active site. Now we can overcome this. So if our cells need to unblock this, right? They need to get this reaction going. Our cells can overcome competitive inhibitors by increasing substrate concentration. Because remember, they're competing. So if there's more of this, then this is more likely to bond than the inhibitors. So we can just, our cells, if we have an inhibitor that's blocking this chemical reaction, well, we just say, okay, well, let's add more substrate to it. Now, non-competitive inhibitors, that doesn't work because non-competitive inhibitors, they don't bind to the active site. So they just sit there. And so they bind to another location and the active site's just not functional. So this cannot be overcome by increasing substrate concentration. If this happens, there's really not a way to stop it except for getting rid of the inhibitor, which sometimes is possible. There are irreversible inhibitors that can permanently bind with covalent bonds. Those would be poisons, okay? So any poisons that you ingest somehow, they usually kill you by becoming non-competitive inhibitors or sometimes competitive inhibitors, but they bond permanently with covalent bonds. They're stuck there forever, and so this enzyme is destroyed, and you can't do an important chemical reaction. So the way to overcome this, the only way to overcome an irreversible inhibitor is to make more enzyme. And if your body can't do that fast enough, you're going to die. So it depends on the poison. If the poison blocks or how much you ingest, right? If you ingest so much that you kill all of these enzymes and these enzymes can't be reproduced fast enough, you're dead. Yeah. So that's why poison kills you, because you change the shape of some important enzymes that your body needs to survive. All right. So which of the following statements about enzyme inhibitors is true? Okay. A competitive inhibitor binds to a location that is separate from the active site. Well, that's not competitive. That's non-competitive. The action of competitive inhibitors may be reversible or irreversible. That sounds true. Let's check the other ones. A non-competitive inhibitor does not change the shape of the active site. That's incorrect. It does. That's why it inhibits it. Inhibition of enzymes by substances that are not substrates can occur only in a laboratory setting. No, because we just talked about poison. So B is going to be your correct answer, right? Okay, let's talk allostery. I know you guys are getting tired. I think we're getting there. I know. I get tired too after a while. Promise. So let's talk about allostery. This is a word to star. We'll talk about allosteric stuff all the time. So allostery is basically what that non-competitive inhibitor stuff was doing. It's binding at a site other than the active site that changes the shape of the active site. And our cells do a lot of this allostery, where we change the shape of things by binding to different parts. So non-competitive inhibitors work through allostery, where they bind to the enzyme and it shifts. Remember that everything works by shape. And if I shift the shape here by binding, I might shift the shape here, which might shift the shape here, might change some of those hydrophobic, hydrophilic things, might change some of those hydrogen bonds, changes the shape of the active site so that it can't bind. Biology. Everything works by shape. So important. Structure determines function. Biology. Shape is key. Shape is key. Okay. And so we can regulate or control our enzymes through allostery. Sometimes our cells need to turn enzymes off, and sometimes our cells need to turn enzymes on. It's kind of like a storage program. If we want to have these in case we need to do something, but we don't want them active all the time, well, then we just turn them on or off, right? Oh, wow, I didn't realize. So this is the active form of the enzyme. Maybe we want to lock the enzyme in an active form. So to do that, we can use an activator, okay, that stabilizes the active form of the enzyme. So you look, this activator just stabilized that active form of that enzyme. But maybe we have this inactive form. Maybe we want to shut this enzyme off, right? So if we want to shut this enzyme off, we can put in an inhibitor. And this inhibitor could be competitive or non-competitive, but an inhibitor will stabilize the inactive form. And so maybe we don't want this enzyme turned on right now. So we need an inhibitor to stabilize the inactive form. But that all works through allostery because it didn't bind to the active site. It bonded to a site other than the active site. So activators stabilize the active form, inhibitors stabilize the inactive form. And it can go back and forth. This is called cooperativity, by the way. Everybody please say cooperativity. Cooperativity is where we can bind at one active site. So if you look, this substrate binds at one active site. So binding of substrate at one active site. Oh my gosh. Locks all active sites in the active form. So if you notice, when I locked this guy into this active site, it changed the shape of the whole enzyme and it locked the other active sites and makes bonding even more likely. So just the bonding of one increases the likelihood of more bonding and makes those go faster, those chemical reactions go faster. Now let's talk about homeostasis and feedback really quick. Okay, feedback inhibition. We talked about feedback loops for a bit. I just can't do it right. Okay. So here, in this case, we are almost done, by the way. I know you're tired. We're so close. The products of the final reaction can inhibit previous reactions. So this is if maybe we want to do a chemical reaction, but we don't want to do it so much that we get too much of a product. Too much of a product can be bad sometimes. So what can happen is we have this metabolic pathway where this enzyme does that reaction, this enzyme does that reaction, and eventually we get the product that we want, right? That product, if we have enough of it, we don't want any more of it, well, that can actually go back and block this enzyme and say, nope, we're done. We don't need any more until we run out. And then when we run out, we can turn that enzyme back on again. This is really important. What's cool about this, though, is usually this ends up being an inhibitor for this enzyme. Like it's usually a non-competitive inhibitor or a competitive inhibitor that blocks the active site. And so the only way to turn this enzyme back on again is to use up all of this. And once it uses up all of this, it'll use the one that's blocking that, and then we can do the reaction again. Holy cow, negative feedback. And it keeps things at stable levels, right? I get too much, it blocks the active site. I run out, I don't get enough, it do the reaction again. Pretty dang cool. I wonder why. Okay. This is really off topic, but I think I need you guys to notice this. Watch me when I write this. Watch me go down. Look what my pen does. Oh, okay, now watch me go up. It's different. Down, up. And then watch when I let go. What the heck? Sorry, distracting. But this keeps things at normal, stable homeostasis levels. We don't get too much of this. We don't get too little of that. So this is an actual example of that, which is pretty cool. So this is an example of threonine, what we want to turn into in the end, isoleucine. So our cells constantly want isoleucine. We need isoleucine. It's an amino acid. So we turn threonine into isoleucine with 5 steps. Holy cow. Told you, it takes a lot of steps. But it turns out that we don't want too much isoleucine. And we do want threonine too. So isoleucine can actually go back to this enzyme here, and it's a non-competitive inhibitor. It binds to the allosteric site, and it blocks it and turns this enzyme off. Now, if we start running out of isoleucine, eventually we'll use this isoleucine up, and we'll turn that enzyme back on, and we can go back to making more isoleucine. So we adjust the output based on the demand, which is so cool. I love this stuff. Okay, which I believe, yep, here's our last slide. I want you to think about the evolution of enzymes, because evolution is a huge topic in here, and how these things evolve is really, really fascinating. So I want you to kind of come up with your own argument to answer these 2 questions. How does an enzyme evolve to survive at a certain temperature or pH? So how do you think that happened, that our enzymes evolved to survive our temperatures and pHs? How does an enzyme evolve to do a certain job? And I know that's a tough question, like really a tough question. I want you to go ahead and I want you to think about that. And I want you to try to answer that to yourselves. How? How do we get that evolution? Okay. Well, guys, this was great. I hope you learned a lot. I'm sorry that was really long. I hope you took that break I told you to take, because holy cow. I can't get my. There we go. Okay. Really quick, I would like you to come meet my sister. My sister's here from California. I want you to meet her really quick. So hold on. That's why I took the day off today and I'm working from home, because I figure if I get to work from home, I get to work with my sister, who's in the other room, and you should meet her. I know this is a video, so you won't be able to meet anybody really, but you can say hi to my sister. Hi. So this is, oh my gosh, I'm so bad at this. This is Brandy. Here. There you go. She's my twin sister. Hi. Yeah. Look, we'll even go like this.
There you go. That's a little easier.
Yeah, she's awesome.
I'm teaching too from my own kids right on the computer. So woohoo!
No. All right, guys. Thanks for sticking with this really long one. Make sure I hope you took breaks. Like I said, see you later. Let's see. And